So yeah, my name is James Bradbury. I'm from the University of Birmingham in the UK, and I'm working as part of the outreach team of Phenomenal. So we're going to start our second session now, which will be Introduction to Metabolites, presented by Kiva Cochrane, who also works at EMBL-EBI. I hope I pronounced your surname correct then.
So hopefully you can see my slides okay. And welcome to this introduction to metabolites session. So my name is Kiva Cochrane. I am the technical officer for the Metabolomics team based at EMBL-EBI, working on the metabolites database. Metabolites is part of the European Molecular Biology Laboratories European Bioinformatics Institute and is accessible via the URL displayed on the screen at the minute. For any queries, you can contact us by email at metabolites-help@ebi.ac.uk. And if you would like some further reading about metabolites, you might like to look at the metabolites publications. The metabolites team at EMBL-EBI has established the metabolites database as one of the most successful international metabolomics repositories. The database is accessible through the metabolites website, which is recommended repository for a number of leading journals, including Nature and the Metabolomics Journal. Our mission is to provide the scientific community with a comprehensive, high quality and freely accessible metabolomics resource. So metabolites hosts data for multiple species collected from various sample types. The study platform includes mass spectrometry, NMR, and imaging techniques, and are currently supported, and open discussion is welcome for all other platforms. So please do get in contact if there's anything else you would like to include. Metabolites benefits from being a manually curated database, which helps to maintain the high quality of its submissions. Our team of developers and curators work collaboratively with submitters to offer added value to their research, providing editorial assistance and support through the submission and annotation process. We are actively involved in promoting metabolomics research through knowledge networks such as Metabolome Exchange and the multi-omics site, the Omics Discovery Index. Metabolites itself is composed of two key areas, the study repository and a compound reference library, which I will talk you through today. So in terms of data sharing, it may not be familiar to you all, especially those coming from non-genomics or metabolomics fields. It's a rare occasion when a biologist might be asked to share an original Western blot image or a plate reader raw data set, even for publications. But the goal for EMBL-EBI is to ensure that the growing body of information from molecular biology and genome research is conveniently accessible to all facets of the European scientific and biotechnology community in ways which promote scientific progress and global competitiveness. The omics generate a vast quantity of data, and metabolomics is no exception. Metabolomics is still a developing field, and what we can interpret today may be greatly expanded tomorrow. Leading from the omics that has come before, we can try to be ahead of the curve, to develop good protocols and reporting systems, to support the integrity of the data produced, to help to develop new tools to add value to the data we are producing, and to make the research available to aid the future development of metabolomics. So what are the benefits of data sharing? Data sharing provides another level of transparency. With clearly defined information requirements, it can support experimental reproducibility. Many recent publications, including this example from the NIH, show increasing awareness of reproducibility issues and development of action plans to combat this. Hence, data sharing is also becoming a more significant part of publication and grant requirements also. Being able to incorporate others' findings into your own research can help both by adding value to your research and reducing costs, helping to generate a new hypothesis. Conversely, if your data set is publicly available, it will be accessible to others who may be developing new tools or looking at new angles, adding further value to your original data set. This will also help lead to future citations and collaborations. With the ideas of data sharing, as the ideas of data sharing have emerged, an urgent need to improve the infrastructure supporting the sharing and reuse of scholarly data has also emerged. A diverse set of stakeholders representing academia, industry, funding agencies, and scholarly publishers have come together to design and jointly endorse a concise and measurable set of principles that we refer to as the FAIR data principles. These put specific emphasis on enhancing the ability of machines to automatically find and use the data, in addition to supporting its reuse by individuals. So examples of this might include using unique identifiers for specific data, using searchable terms which can be searched by person or machine, using data associated with their provenance, making it traceability, clear and accessible metadata as well, and using controlled vocabularies. Here we can see an example of unique identifiers. The metabolite studies are each given a reference identifier beginning MTBLS. We can see this at the bottom of the screen. So this particular study is MTBLS1. Another method which helps to aid data traceability and interoperability is the use of controlled vocabularies on ontologies. So why is this important? If we take, for example, the data displayed, the example displayed for MTBLS1, 100 people were asked to name the species used in this metabolite study, to which 8 different responses were noticed. Each of these terms may bring back different results if used in a search or text mine. However, if we actively use a controlled vocabulary in order to manage our data, we will reduce this variety. For example, using the official taxonomy for all species information, in this case Homo sapiens, will greatly improve our traceability. So let's have a look at metabolites itself. There are various ways to find information through the Metabolites website. Starting from the main page, we can look at the options now of how to browse and search metabolites. You can browse areas of interest, including studies, compounds, and by species. We can also, within the Browse Species option, you can use our model organism links to find studies and metabolites. You can also use the taxonomy guide to find information related to species, selecting for different classifications. And we can also use the search term, which is located in the top right-hand corner. You'll find the search box, and you can just enter any specific search term you would like to look for. Within the results sections, information can then be filtered further according to, for example, the instrument or sample type, using the expandable boxes on the left-hand side of the screen. So, looking specifically at the study repository, we want to consider why we should be sharing data first. The Metabolites database requirements have been guided by the Metabolomics Standards Initiative guidelines, which were part of the initiative set up by the Metabolomics Society. The early efforts of the Metabolomics Society Standards Initiative were focused on community-agreed reporting standards, the so-called minimal information checklists and data exchange formats. The Metabolomics Standards Initiative, its aim was to provide a clear description of the biological system studied and all components of the metabolomics study, as well as to allow data to be efficiently applied, shared, and reused. So 5 working groups were initially established. For example, the chemical analysis group proposed minimum information for reporting chemical analysis, including minimum metadata, reporting related to metabolite identification. Therefore, several cross-communicating working groups were built to cover the development of different aspects of metabolomics experiment data standardization. These guidelines provided reporting standards for all stages of metabolomics analysis: experimental design, biological context, chemical analysis, and data processing. Since the guidelines were developed, a series of public metabolomics databases and repositories which accept the deposition of metabolomic data sets have arisen. Public studies on the metabolites database are also shared with Metabolomics Exchange, an initiative set up to provide an aggregation and notification service for the metabolomics community. The guidelines also include a method of assigning the confidence level of metabolite identification based on the experimental analysis type. Here you can see a confidence level of 1 to 5, with level 5 being an exact mass match, and level 1 being a confirmed structure. The first guidelines were developed around 10 years ago, and as of 2018 are now under review. MetaboLights has used the guidelines to develop a data management method for metabolite study deposition. So as described in the beginning, metabolites host a variety of information. Examples given here include different sample species and techniques. Some studies are interested in using data to develop new analysis tools. Another useful feature that you will find when you search metabolite studies will be tags which link groups of studies together. So for example, at the bottom here, we have the malaria host pathogen interaction center with the tag at the bottom of the title, which will then link through to all studies associated with this group. Looking at the information collected for studies, we can see an example here, which is MTBLS 100. So again, as described in the beginning, we have sorry, I'll just move that out of the way. We have options to look for studies in particular. So we have the search. We can put in a direct search for a study name, or we can do this through the browse study features and then the filtering at the side. So within this, once we have our results, you will see, first of all, the title of the study, and then you will see further information about the study, which includes a unique identifier for the metabolite study, MTBLS, with a number following it, information about the study size and the submitter, and also some key information about the study, including the organism and the specific study factors. Clicking on the study title will then take us through into the study itself. So the first page you will see is the investigation file. At the top, you will notice a status bar. When you first submit your study to MetaboLights, this will be highlighted only to the submitted section. As your study progresses through curation and review, you will see more of this status bar being highlighted until finally, when the study becomes public, the whole bar will be highlighted following your chosen date for publication. The next section gives a brief abstract about the study with quick links to the metabolites identified and a download for the metadata files. For those of you who have an ORCID ID, there is also an option to claim your study. Following this, there is a section of tabs that will guide us through the study information. So in this first tab, we see the study design description. Here you will find an overview of the species, techniques, and key factors. You will also find the link to any publication associated with the study deposition. Moving across, you will find all the information related to the experimental protocols, where samples came from and how they were extracted, through to how they were analysed, and finally what methods were used to identify the metabolites themselves. Specific text mineable information is gathered on the following tabs. So for example, in the sample sheet tab, you will find the information in columns relating to organism and organism part, with some clear sample name identifiers. You may also find in this section some further columns known as study factors, where you might like to include further information about your samples. For example, age, if it's related to human samples, sample collection times, or different types of sample treatments. In the next assay, you will find two main sources of information. The first is the all-important metabolites identified within the study. This field gives information about the peaks identified in each sample as you scroll across to the right. Where metabolites have been identified, the compound names are provided, and they are assigned to a specific ID using the EBI chemistry ontology. This is again a controlled ontology, which means that the metabolites that you're looking for can be easily identified with linked information. The EBI links and EBI IDs here are clickable links, which will take you to further information about your particular metabolite. Also provided are the INCHI and SMILES for structural information convenience. In the lower half of this tab, you will also find further information about the assay. So if I draw your attention to the top of the field, you will see under assay, you will be able to see the particular technology that was used. In this case, it's NMR. If this is mass spec, you will see this here also. And then in this lower section under data, you will see the more specific text mineable version of the assays. So within this section, you may find information relating to specific chromatography or mass spectrometry equipments, particular types of columns that were used. In this case, this is an NMR example, so you can see that it was a hydrogen labeled NMR spectroscopy. You can see the size of tubes that were used, the solvent, pH, temperature, and various other bits of information that are important to being able to reuse the data produced. In the next study tab, we'll find the study files. Within this, you will find both the metadata information, which is the information which is uploaded, including the protocols, the abstract, the background information about samples, and also the direct raw data files. You have a few options here. You can look for particular files using this list of studies field, using say .txt or .zip to search for particular file types. You can then opt to download studies if you would like to reuse the data. So there is the option to download study files in this top right corner. There is also along the central section, an option to download the whole study via different methods, so FAST and FTP, or to just download the metadata. You can select specific files to download by clicking the boxes along the left-hand side, and then at the bottom of the file, selecting to download the selected files only. In the next tab, you can then see the study validation. So in this case, as the study is public, it has passed all of our validation criteria, and you'll see this nice green tick at the side, and the status is that it passes for each of the different required fields. If you are submitting your study, you may see a variety of different options as you come to your study submission. Firstly, in the study validation tab, you can see on this screen, there is an X right beside study validation, which means that the study is not complete and is not ready to be curated. Whereas in this screen, you can see that the study validation has a tick, which means it has passed the curation stage. So within this validation, you can see if there's an X, it tells you that it fails, and it will tell you information about why it isn't failing, or if it's an orange X, it's incomplete. So this might be that you have a publication, but it's not ready yet, so you haven't provided the full information, but it will be provided at a later date. So this is important to note when you're actually submitting studies to MetaboLights, how your validation is actually going. And finally, we have the tab, which is the Pathways tab. And this uses the metabolites identified within the study. And these are exported to MetExplore, which then can provide a different interface to provide you with potential pathways related to the metabolites which have been identified in the study. So you can see examples of an interactive network analysis, and at the bottom of the tab will be lists of specific pathways which you can then click on to look at in further detail. So next we're going to look at a bit about the compound reference. So within this section, again, we're able to browse compounds or search for specific compounds. The results will be produced, and we will have a filter option along the left-hand side. Slightly different from the study filter, you will be able to then select specifically if you want a compound that has information related to species, pathways, reactions, or for NMR or mass spec. So here is an example of a compound reference. We have oleic acid. You can see at the top we have the compound name and the specific metabolites compound identifier. And then we have another series of tabs that take us through the information about this compound. So starting with the chemistry tab, you can see that on the left-hand side, we will have structural display, which will also will show you the structure and also give you links to the mole, smiles, and inchi files, which you can download and use. We also have some of the chemistry-specific information available as well. We then have some biology-based information. This information is based on looking at oleic acid specifically and links that come through. So you can see for different species, we then have information about different publications or about metabolite studies that are available related to oleic acid. And here you can see that this publication reference will then take you to this particular paper. In the next section, we can then look at pathways that oleic acid is actually involved in. There is a few different options for these pathways. We can look using wiki pathways. This allows us to select for particular species and then particular types of pathways that you might be interested in. We have the option to look at the KEGG pathways, again, looking at the different specific types of pathways, and also at reactome pathways, selecting for species and the different areas of interest. These are drop-down menus, which will then give you other options for different pathways of interest. So moving on from that, we will then have spectra option. So at the top of this, you will have the select mass spec spectra. So these are different spectra that have been deposited in metabolites. And we encourage you that if you have good spectra for specific metabolites, to please think about depositing with us. You can do this through the upload spectra section on the top right-hand side here. And otherwise, you can contact us directly if you would like to talk about uploading some of your spectra. So there's different options depending on where the spectra has come from. We have a spectral viewer, and then at the bottom we have further metadata about this particular spectra. In the next tab, we then have the reaction links, so reactions specific that oleic acid is involved in. These are linked to the RHEA database, and again there is a drop-down menu which allows you to select your specific reactions of interest, which is then displayed in the screen below. Again, this is the drop-down menu with the various reactions that oleic acid is involved in. And finally, we have literature links. These are specific links that have come from the KEBI database for biological references to oleic acid. Each link can then give you more information and will give you a brief abstract background to the literature. Within all of this section, we are currently developing further features for this, so look out for more information coming soon. So metabolite submission, in terms of the submission pipeline, we use ISA Creator as a third-party platform. The study is created in ISA Creator, uploaded to MetaboLights, and then we can then send you a reviewer link in order for you to share this pre-publication in a private setting. And once your study is published, or once your publication is available, we can then create or make your study public. So ISA Creator is a software suite which was designed specifically for biological data to be stored so that the resulting data and discoveries are reproducible and reusable. We use this in the background. It allows you to create an investigation. Within the investigation, you will have a study. And within the study, you can have multiple assays. So you could have your positive mass spec assay with your negative mass spec assay, for example. And then this provides further information. It will link further information, including the raw data background. So The ISA Creator package, complete with the Metabolites specific package, is downloadable from the Metabolites site. And further information and guidance for using this is available through Metabolites Help, with various different topics to help you. When you open the ISA Creator, you will see an option for ISA Creator Lite or ISA Creator. Either are usable. You can create a username and password to log on, and then you'll be able to use the Metabolites configuration to actually start your Metabolite study. Within this, you will see 3 main tabs. So this is your investigation tab, your sample definition tab, and your assay tab. And within the assay tab, you will also find a link, if you scroll to the far right-hand side, for the metabolite assignment file, which will open up a file where you can put in all of your metabolite identifications. So what you will find is that each of these tabs are directly linked to the metabolite studies on the website. So you can easily look at the website for further information on what should be stored within the different sections of the ISA Creator tabs. So you can see here that this is the investigation or study tab field, the first tab on ISA Creator, and it links to the study design description and protocols within the Metabolites site. Then we have the sample information, again linking through to the tab, and the assay information linking through to the assay tab within Metabolites. In order to submit the study to Metabolites, once you have finished your ISA Creator study, what you will do is you will zip the ISA Creator files together with your raw data files, which you will have provided links to within ISA Creator. And then you will come to the Metabolites website, create an account, log in, and go to submit a new study, either in the top right or here. So finally, coming soon to Metabolites, there is a need to bridge the gap between data deposition platforms such as Metabolites and the analysis platforms such as the phenomenal Galaxy workflow that you're learning about today. In order to do so, we have created the Metabolites Labs workspace. This will allow you to create your own Metabolites workspace, which you can upload your raw data to, and then transfer it either to a workflow to do your analysis or directly into the Metabolites database. This tool will also allow you to generate some of the ISA Creator files without having to manually curate them. So there is an option within it to use the NMR ML to ISA or MZ ML to ISA to directly pull the information from your raw data files and put them into the Metabolites submission files. So what does Metabolites Labs look like? Here you have an example of a project space. You can create a new project. You will have 2 options. You can create a brand new project and upload your own information, or you can clone an existing study within Metabolites should you want to use the raw data to reanalyze. Here you can see it's an easy upload for your data. This is the process, and here are all the files uploaded within the project set here. You can see for exporting to Metabolites on the right-hand side, this green button for MZ-ML to ISA. So it is aware that it is a mass spec files, so therefore it only gives the MZ-ML to ISA option. If it was NMR, you would see that option, and then you can export to Metabolites here. So this is currently available in the beta version. If you sign up to Metabolites and log into Workspace, you will find access to MetaboLights labs. We are really interested in getting feedback from users, so please have a look and please let us know what you think. Also coming soon, we have a brand new website which will have all of the same information I've talked you through in a much more user-friendly display. We also have new online study editor. So although there will still be some need for ISA creator use, much more of the information will be directly editable online. We have a public API, which will be available for those of you who are coming from service companies or phenome centers, which might help with the process of uploading multiple studies directly to MetaboLights. And to go with all of this, we will have new training material coming soon. So for further information, you can see MetaboLights training at the EBI site. The URL's at the bottom here, EBI training/online. You can also find us at www.ebi.ac.uk/metabolites. You can email us or follow us on Twitter. And you will also find us at the upcoming Metabolomics conference in Seattle. We have a presentation in the Big Data, Databases and Cloud Computing section, and also a poster. So please come and find us and have a chat. So that's me complete. Thank you very much.
Great, thank you, Kiva. So if there's no questions, then I'd like to thank everybody for joining today's webinar, and we look forward to tomorrow's sessions. Thank you.